Good evening. I'm Dick Meserve. I'm the president of the Carnegie Institution, and I would like to welcome you all to a Capital Science evening. 150 years ago, people had no idea what determined the sex of a baby. Some believed the emotional state of the pregnant woman determined whether her child was male or female. Others, more scientifically inclined, thought sex was determined solely by the properties of the egg. The temperature of the egg, the food available to it, or even the centrifugal forces acting on it were thought to play critical roles. That chromosomes might be involved in sex determination was an idea first considered late in the 1800s, nearly half a century after chromosomes were first identified. But it wasn't until 1905 that two researchers, Edmund Wilson and Nettie Stevens, independently discovered that in some insect species, chromosomes are different in males and females. Nettie Stevens, working at Bryn Mawr College and supported by the Carnegie Institution, identified the Y chromosome in the mealworm and deduced that the chromosomal basis of sex depended on its presence or absence. Her important work, published as a Carnegie monograph, was to become central in subsequent discoveries confirming the chromosomal basis of heredity. The Y, or male chromosome, was a latecomer to the science of genetics. It was one of the last of the chromosomes to be identified. That it took so long is not surprising. In most species, the Y is tiny. In humans, it contains fewer than 50 working genes, compared to over 1,500 on the X chromosome. In 1905, Edmund Wilson noted that the Y was so small in some species that it seemed in danger of disappearing. Little did Wilson know how true his remark actually was, at least in the case of humans. Over evolutionary time, the Y chromosome in humans has shed many of its genes, shrinking to a shadow of its former self. As our guest tonight, Dr. Jenny Graves, will tell us, the human Y chromosome is indeed in danger of disappearing. Dr. Jennifer Ann Marshall-Graves is a professor of comparative genomics at the Research School of Biology of the Australian National University in Canberra. She is also the director of the interdisciplinary ARC Center of Excellence for Kangaroo Genomics, a collaborative effort involving several universities and research centers. Her comparative genomics approach, using kangaroos and other mammals unique to Australia, has had a major effect on current thinking in the field of mammalian genomics, particularly in the evolution of the sex chromosomes. Her discoveries, including the discovery of the human sex-determining gene, have deepened our understanding of sex differentiation and led to insights not only in mammalian development and reproduction, but also into diagnoses of gender disorders and gender-related human diseases. A native Australian, Dr. Graves received her BSc and her MSc degrees from Adelaide University. In 1971, she received her PhD in molecular biology at the University of California at Berkeley. She lectured at La Trobe University in Melbourne for 30 years before moving to the Australian National University in 2001. Dr. Graves was elected a Fellow of the Australian Academy of Science in 1999, and she is now the Academy's Foreign Secretary. In 2002, she was awarded a Centenary Medal for services to Australian genetics and genomics by the Australian government. In 2006, she received the Macfarlane Burnet Medal for Biology by the Australian Academy of Science, and in 2006, she was elected the Asia Pacific Laureate of the L'Oreal UNESCO Awards for Women in Science. Dr. Graves is an inspiring scientist and teacher, has deepened our understanding of the great web of life. In doing so, she has launched a debate about the future of evolution of our species. Please join me in welcoming Dr. Jenny Graves.
Well, thank you very much, Dr. Meserve, and it's a real honour to be invited to lecture at the Carnegie Institution in this gorgeous auditorium and in such a very inspiring series of lectures. Obviously, I'm working on sex determination, but I'm interested in many other aspects of human genomics. And what I'm first of all going to tell you about is how sex is determined in humans. What I really want to tell you is how peculiar sex chromosomes are. And then I'll talk about how they evolved to be so peculiar. I'll say a little bit about their origin using my research not only in kangaroos, but also other weird mammals and reptiles as well. Then I'll talk about the future of the XY system, particularly the degradation of the Y chromosome, and finish up talking about the future of men. And my subliminal message here is going to be how valuable weird animals are, because they can tell us something we didn't know about ourselves. So the first weird animals I want you to meet are the animals in my own lab, and there are 3 there who are named who've been very important in my story. This was taken several years ago when I was still at La Trobe University, but they set the stage for a lot of work that's happened subsequently. But what I want you to notice is that men and women really are different. They're phenotypically different. You know, the men are usually taller and bigger and stronger, and the women are less so. Everything is, of course, averaged, and there are always exceptions. But what I usually do, because this subject is so controversial, is often at meetings like this, I'll hand out cards and invite the audience to write down as quickly as possible all the differences they can think of between men and women. And that's what this is. This is actually comes from a high school class a number of years ago, but in fact I got exactly the same answers from the Royal Society of New Zealand. So one thing that nobody mentioned was the really important difference between men and women, and that is that they make different kinds of gametes, eggs and sperm. What people were much more interested in was the reproductive equipment. And I have to tell you that breasts and penises won hands down. But the other thing that people always notice are the secondary sexual characteristics, the difference in body build and hair and so forth, and particularly they are interested in the behaviour. Now remember, this is not me saying that men are loud and silly and women are calmer and submissive. This is a Year 10 class, but you know, overall there are behavioural differences and physical differences as well as reproductive differences. So there are a lot of differences that people notice. And of course, what you probably don't know is that there's only one difference that is really meaningful, and that is that men have testicles and women don't. Everything else flows from that, as I'll explain. So what happens after about 12 weeks after a baby is conceived, you really can't tell which are going to be boys and which are going to be girls. There's just this ridge of cells called the indifferent gonad that hasn't decided yet whether it's going to be a testis or an ovary, and then it gets some kind of a signal that we call the testis determining factor, and that turns it into a testis in the embryo, and that churns out hormones, and it's the hormones that make you male and control all those other characteristics. If you don't become male, some weeks later that ridge of cells turns it into an ovary and the effect is a female. So we know that's what happens, and that's been known for a long time. And the question then is, well, where is the testis determining factor, and what is it and what does it do? So that's what I'll tell you a bit about now. You probably all know that males and females have 46 chromosomes each. That's 23 pairs. And what can be done is to cut these out of the photograph and line them all up. So you can see that there's 2 copies of each chromosome in a female, one from your mother, one from your father. And males are the same except for one chromosome at the end. So we call these ordinary chromosomes autosomes, that is, other chromosomes. But this one chromosome, which is called the X chromosome, is present in 2 copies in females and only 1 copy in males. And the reason it's actually called an X chromosome is not because it's X-shaped, as many people think, but because it's X for unknown. But males have another chromosome that's called the Y, as Dr. Meserve said, and it's a very little chromosome, but it's very important. So I'll tell you a bit about those chromosomes. So if we blow them up, you can see that the X chromosome is a very decent sort of chromosome. It's a very ordinary kind of chromosome, lots of genes on it. And the Y is really a pathetic little thing. And if we look at the genes on it, we can see that at the last count, there was something like 1,340 genes on the X chromosome. They do all kinds of different things, and they're widely expressed in all kinds of tissues. But the Y chromosome, there are only 45 genes that actually make a sensible protein, and most of these are specific to the testes. So it's a very male-oriented chromosome. So that's not very many genes, but one is extremely important, and that's the TDF gene, because that lives on the Y chromosome. And what it does is it turns on other genes that then go to make a testis, and it's the testis that makes hormones, and hormones that make you male. So we know that that's how it worked, and there was a huge effort to try and find out, well, what is this testis determining factor? And this is really where I came in, because I didn't work on sex at all, until there's a claim that this gene was the first gene that was cloned from the human Y chromosome, which is called ZFY, for zinc finger gene on the Y chromosome. And that's when I was asked if I could show that the ZFY gene is on the Y chromosome in kangaroos as well as in humans and mice. So what we did was we cloned that gene and we mapped it in marsupials, and the answer is it wasn't. It's the wrong gene. And that work was done by Andrew Sinclair, the guy in the red braces there. And he was the one who went to London and worked with Peter Goodfellow to clone the right gene, which is called SRY. And my other student, Jamie Foster, was involved in that work too, quite by accident. He happened to be on holiday in England, and I said, well, why don't you go and see Andrew? So he stayed there for a year and helped clone SRY, then came back and cloned another gene that we didn't suspect was there, and we accidentally cloned it on the X chromosome. And it's clearly the gene that evolved into SRY. And I'll say a bit more about that later. So getting back to sex chromosomes, there they are, the big glorious X chromosome and the pathetic little Y chromosome, but with its SRY gene that does it all. But sex chromosomes, when you think about it, are nothing but trouble, because they don't pair very well. They only pair at the very, very tip, and so there's a lot of problems at meiosis when they don't segregate properly. There's also problems because women have 2 copies of the X and males have only got 1. And what that means is that there's twice as much many copies of genes on the X in females than males, and there has to be a compensation mechanism has evolved to take care of that, which is very, very complicated. There's also sex-linked diseases because men have only got 1 copy of the X. Any mutation of an X gene is going to have an effect, so that's why haemophiliacs are almost always males, and there's many, many diseases that are pretty much male-specific because it's a gene on the X which mutates. And also sex reversal happens quite a lot because the SRY gene accidentally gets swapped onto another chromosome. So you have to ask, well, why are they so weird? Is it because that's the way they work optimally, or is it some horrible evolutionary accident? And I'm here to tell you that they do not work optimally, and they are indeed a horrible evolutionary accident. And this is, in fact, a lovely example of what I call dumb design. So the reason sex chromosomes are the way they are is because that's how they evolved. And we know that what has happened here is that once upon a time, the X chromosomes were really just an ordinary pair of chromosomes, until one day, one partner acquired a new male-determining locus. And that was the kiss of death for that chromosome, because what happened next was that other genes that are important in males clustered around the sex-determining locus, and because all those genes had to be kept together, that chromosome stopped recombining with the X chromosome. Now, that's a very bad thing to happen to any part of the genome because it can't repair itself. And so what happens then is you get a lot of deletions and mutations in that region. And so that region spreads and spreads, and you lose active genes and you import junk, and you end up with chromosomes that look pretty much like that only a little tiny bit at the top pairs with the X chromosome. But this process can go on until the Y chromosome is completely isolated and doesn't recombine at all with the X. And you can imagine it could even disappear. Well, what's the evidence for this? In humans, it's very clear when you look at it because, in fact, although those chromosomes look completely different, there's a lot of homology. And so most genes that are on the Y chromosome actually have friends on the X chromosome from which they obviously evolved. And so clearly the Y chromosome is just a degraded X chromosome. So, well, what does that mean as far as what genes are on the X and the Y chromosomes? The X chromosome is interesting because it's very, very conserved. If you've seen one, you've seen them all. The human X chromosome is almost identical to the elephant X chromosome. And it's got lots of genes and they do many different things and they have many different functions. So we always used to think, well, that's because essentially it's the same, nothing much has happened to the X since the olden days in which it was an autosome. It's just as sweet as it always was, and that leads me to my models of the X chromosome, and this is the immaculate X chromosome that nothing has happened to at all. But now we have the genome, we know that's not true. In fact, there are a lot of genes on the X chromosome, too many genes that control other genes. And there's also too many genes that are expressed in the brain and seem to have something to do with intelligence, and that's why there's so much X-linked mental retardation. It's largely boys who have mental retardation. And also the X chromosome is a very sexy chromosome because there's far too many genes on it that have something to do with reproduction, particularly male reproduction. And the really weird thing is that there's a lot of genes that do both. And I call these brains and balls genes, because they're expressed both in the brain and the testis, and it's always been a real question as to why so many genes have both functions. So why then are there so many genes on the X chromosome that are involved in sex and in intelligence? Well, in fact, people thought about this back in the 1930s, and the answer seemed to be, well, you might expect male advantage genes to accumulate on the X. And I'll just show you why. If, for instance, you've got a gene on one of the autosomes, so that means there's 2 copies in both males and females, and this gene, for instance, has some critical function in making sperm. If then you have some kind of mutation that makes the sperm bigger or faster or more sperm, that's not going to have any kind of effect if that mutation is recessive, which most mutations are. Of course, females don't make sperm anyway, but even in males, you won't get an effect unless both of those genes are mutated. But look what happens if you're talking about the sex chromosomes, the 2 X in female and the single X in the male. Now you can see there's no effect in females, but there's an instant effect in males. And so that means that if there's a mutation that arises on the X chromosome that's an advantage for males, it'll be immediately seized on in males. And that's true even if it's bad for females. So you get what we call sexually antagonistic genes accumulating on the X chromosome. So this male is going to make bigger, faster, fatter sperm, and that X chromosome is going to be immediately selected very strongly. So it's not surprising then that male reproduction genes are on the X chromosome, but what about intelligence genes? I mean, is intelligence a male advantage trait? I don't think so. Is there, for instance, a big brain mutant that acts in the same way? And the best hypothesis I've heard is that what's happening here is that females are selecting smart males. So they may select smart males just because they bring home more mammoth steaks, or they may select smart males for the same reason that the peahen selects a peacock with a bigger tail, because that's a badge of good genes. So this is what we call sexual selection, and it's very, very rapid. So it could explain why the human brain doubled in size in just a couple of million years. Well, what about the brains and balls genes? Brain and testis is not, they're not very similar tissues. So, but you can see many, many genes that have an effect of mutation both on the gonads or on fertility and also intelligence. So many mental retardation syndromes are accompanied by something wrong with the gonads. And people have wondered for years and years why this might be. And what I want to suggest to you is that we're looking at the wrong thing. If we're looking for a functional link, we're probably looking for the wrong thing. We should be looking for an evolutionary link. So what I suggest is that the raw material for the selection was some kind of really useful protein, a multifunctional protein that does all kinds of different things. And it's subject, once it gets onto the X chromosome, it becomes subject to direct selection for a function in male reproduction. So, for instance, it may become selected for making bigger sperm, for instance, by being targeted to the testis. But at the same time, it's under selection, sexual selection for intelligence. So the same protein may then do quite different functions in the two different organisms. And I think this might be a lovely example of how you often will ask a functional question, but you'll get an evolutionary answer. So I want to start off talking now about the Y chromosome, and as I've said before, the Y chromosome is mostly junk. There are only a few genes, and most of these are actually shared with the X chromosome, but these genes mostly have male-specific functions. And the other thing about the Y, completely the opposite from the X, is it's very poorly conserved. So here's my models of the Y chromosome. The one on the left is the one we all grew up with. The Y might be very small, but it's a macho little thing and it's very powerful. But in fact, we know that the only reason for that is because it contains an SRY gene. There's also a model in the middle that the Y is actually a selfish element, it's sort of grabbing handy genes from other places in the genome that are good for male reproduction. But my favourite model is the one on the right. The Y is simply a wimp. It's just a pale shadow of its former glorious self, which of course is the X chromosome. So people have written books about why the Y chromosome degrades, and I really just tell you that there's two reasons. Everything's against the Y. One is it's always in a testis. By definition, it's never in an ovary, any generation. And it turns out the testis is a very dangerous place to be because there's many rounds of cell division, so many opportunities for mutation, and very poor DNA repair. And so there's very high variation of the Y chromosome because it's always in a testis, whereas other chromosomes are only in a testis in 1 out of every 2 generations. But the other thing is that selection doesn't work very well. And that is because it doesn't recombine. So you're not selecting just for one gene, you're selecting for a whole Y chromosome. So if you're selecting for a gene, for instance, that's mutated into making bigger, faster, fatter sperm, but you'll get whatever else is on that particular Y chromosome, good or bad. And so that's called a hitchhiker effect, and it means that the Y chromosome degenerates very rapidly. So sexually antagonistic genes are very interesting because they're genes that are good in one sex and bad in another. And you can see why this might be so, because the interests of the male genome are entirely in having this particular baby with his genes, and it's got to be the biggest and toughest baby. And it doesn't matter what it costs the female, because there are plenty of females around. But of course the female wants to survive, to have more offspring. So this is what sets up an antagonistic situation. And what we can see now is that sex chromosomes are very good places to put these sexually antagonistic genes on. The X chromosome has male-specific genes on, and that's great because males only have one, so they express it, and females won't express it until it becomes very frequent in the population. And of course, being on the Y chromosome is no problem because only males have a Y. Well, let's talk about genes on the Y chromosome. These just happen to be 3 genes that we've worked on. The SRY gene I've already mentioned. There's a spermatogenesis gene called RBMY that we know is required for making sperm because men who have a deletion are infertile. And there's another gene called TSPY, and this is typical of many genes on the Y chromosome. So it turns out that all of these have copies on the X chromosome from which they obviously evolved, and these just happen to be the 3 that we discovered. And they're a very interesting bunch of genes because many of them have brains and balls genes. So they're expressed in the brain, and deletion will give you mental retardation, and many of them are expressed also in the testes, and they affect spermatogenesis. So they're widely expressed when they're on the X chromosome, but they become testis limited when they're on the Y, and they are, as I said, these brains and balls genes. So what it seems to be is most of these genes are actually relics of genes that were on the X chromosome to begin with. So we've been very interested in having a look at, well, how did RBMX evolve into RBMY and change its function? And it looks like it probably just changes. It's a small change and now has a different binding partner, so it's expressed exclusively in the testis. So most work has been done on how did SRY evolve, and I just want to use that as an example. So if we look at the SOX3 gene, and we know that that was the ancestor of SRY because it's the most similar to SRY, we can look at that gene and find out, well, where is it expressed? And it's a typical brains and balls gene, so it's expressed in the brain and the central nervous system, and deletion makes you mentally retarded, but it's also expressed in the testis, and deletion makes you infertile. And it's quite a big gene, and it shares that little bit in the middle, which is called an HMG box, and it's a signature for a whole family of genes, some of which are very important in development. And its role is to bind to particular sites of DNA and bend them into a particular angle. And all these genes have the same effect of binding somewhere and bending DNA and somehow rather affecting its transcription. So when we had a look at that gene and compared it to the gene it became, the SRY gene, this was a big surprise because most of the gene has disappeared completely. So it's only the box which is there, and it's got stuck together with all kinds of other factors, transactivating factors and binding factors, and they're actually rather different in different species. So, for instance, the mouse SRY gene has that long green tail on it, which is a single amino acid practically, and that's not on any other genes of primates, for instance. It's rodent-specific, but it's become necessary because if you take it off, that gene won't work anymore. So it's a very surprising gene, but it seems to be essentially just that box which binds to and bends DNA, and somehow that activates other genes in the testis-determining pathway. Well, what about how these genes on the Y chromosome are selected for a male determining function? Well, that's easy to explain because, again, if we look at the autosome and a recessive mutation on autosome that makes bigger, fatter sperm is not going to have any effect. But if it's on the Y, it will have an immediate effect on the male, no effect at all on the female. So obviously this is a very good place for sexually antagonistic genes. This seems to be why all those genes that were once upon a time on the X chromosome and had rather general functions have been evolved to have a male-specific function in the testis. Well, the other surprising thing about the Y chromosome is that if you look at the Y chromosomes of different mammals, they're very different. And that's quite unusual because practically the whole genome of a mammal is extremely conserved. You've probably heard it said that the human genome is almost identical to the chimpanzee genome. Essentially the same genes doing the same job and in the same place in the same order. But even if you look at the elephant genome, it's pretty much the same set of genes doing the same job. Occasionally you'll find a few extra copies of this or a few extra copies of that. But you don't very frequently get completely novel genes arising in any lineage. So the Y chromosome is very surprising. So there's the human Y chromosome, there's a whole bunch of genes on it, and if you look at the mouse Y chromosome and the cow Y chromosome, you can see that they're a different set of genes. So the ones in dark blue are the ones that are active, the ones in light blue are no longer active, and the ones in white are gone. The interesting thing is that these all relate to the X chromosome. So they all have a partner on the X chromosome from which they evolved. So what it looks like is that independently in different lineages, different genes have dropped out. So, for instance, you can see that there's a gene called UBE1Y, and that's associated with UBE1X on the X chromosome, but in primates that gene's gone. But in mouse, it's actually rather important for fertility, and it's still there in cows as well. So it looks like in each lineage you've had dropping out of different sets of genes, including genes that we know to actually be doing something useful for the male. So this is what led me to predict that what's going on here is that genes are being lost progressively from the Y chromosome. So starting from something which was identical to the X chromosome, you start off with the set of genes that actually does have a role, but one by one these genes drop out. Now you can't just lose genes, so presumably what's happening is that other genes are evolving to take their place. And many of these are members of gene families, so we think probably other members of the same family take over their function in spermatogenesis. So you can imagine that this Y chromosome is getting smaller and smaller, and eventually it's really only going to have the SRY gene left, and if that goes, well, is the Y doomed to disappear? And there it goes. Well, I'll talk now about the origin of the XY system, and that's my chance to tell you a bit about the amazing animals that we work with, because we've been able to chart the origin by comparing the Y chromosomes of humans and mice with other mammals. So far I've only talked about humans and mice and maybe elephants, and these are what we call placental mammals. But they're not the only mammals there are. There's also marsupial mammals. And marsupial mammals shared a common ancestor with us about 148 million years ago. And then there's another group of mammals called the monotremes, and we shared a common ancestor with them 166 million years ago. So if we want to look back in time at the sex chromosomes, we just take the sex chromosomes of marsupials and monotremes, and we can figure out, well, what did you look like 148 million years ago, or 166 million years ago, or 310 million years ago, and that's when we shared a common ancestor with birds and reptiles, or even longer ago when we shared a common ancestor with frogs and fish. So you probably know that we now are able to sequence the entire genome of numbers of animals, and you'll see by my dots here, the big dots are for genomes that have been sequenced very thoroughly, and little dots are for genomes that have been sequenced very lightly. So there's been a lot of activity sequencing the human genome and the chimp genome and mouse and rat and cats and dogs and even elephants now. And there's also been quite a lot of activity sequencing the chicken and other birds and a frog and several fish. But you can see that there's a great big gap in the middle, and you can say, well, wouldn't it be lovely if we had something in that gap? And we do, because we have monotremes and marsupials fill this gap very nicely. So they've ended up being very important, and it's been possible to do the genomes of both the platypus and opossum. Those were cover stories in Nature in the last couple of years, and we're now doing the kangaroo genome. And more recently, there's a little creature called a Tasmanian devil, which has become very important, and we're now sequencing that as well. So it would be very interesting then to have a look at the sex chromosomes of marsupials. We've done that for many years. What we wanted to do was to look at the marsupial genome, and we made an application to sequence the kangaroo. Unfortunately, NIH decided it had to be an American marsupial, so they chose the opossum, the Brazilian opossum, mind you. And that was published a few years ago, and we're now doing the Tasmanian devil. The reason we're doing the Tasmanian devil is it's quite distantly related from marsupials, but also because devils are in diabolical trouble because they're getting this horrible transmissible cancer that makes a terrible mess of their face. And it's likely that this species will become extinct in about 10 years because this cancer can actually be transmitted from animal to animal. So we just published the transcriptome of the animal and the genes that are transcribed in the tumour. So one thing I have to tell you is that marsupials have absolutely gorgeous chromosomes. They're so gorgeous they actually made a stamp out of them a few years ago. And the other thing is that if you've seen one, you've seen them all. They're very similar and they're related to each other very simply. And this lovely technique allows us to see this because we can actually sort chromosomes out by size using a fluorescence activated cell sorter, and then we can make DNA from an individual chromosome and we can tag it with a particular fluorochrome. So here's a Tamil wallaby, a chromosome 2 DNA tagged green and a chromosome 7 tagged pink and the X tagged white, and we can then throw those paints onto the chromosomes of another species. This is another wallaby species, and the chromosomes look quite different, but you can say it's the same old blocks of genes that have just been put together in different ways. And this is true of all marsupials, so it means that if we have a map and a sequence of one, we can very easily infer what it is in another. So kangaroos really do look like this. They lounge around practically the whole day. They also, like humans, have an XX female, XY male system. And you can see that if you look at their real live chromosomes, as you can see they're simply gorgeous, and line them up so there's two, one from mum and one from dad, in everything except the X and the Y, which are rather small chromosomes as a matter of fact. But the question is, well, is it the same X and Y or is it completely different? So we did a lot of mapping of genes, which we can easily do by cloning the gene and then tagging the clone with a fluorochrome and then looking to see where it goes to in the chromosome. So here's two genes tagged in different colours. They're very close to each other on the human X, and you can see they're very close to each other also on the kangaroo X. So we've mapped the whole X really quite easily, and what we found, which was a great surprise then, was that if this is a picture of the human X chromosome and the middle bit is the telomere, we can see that all the genes in that gold region are on the X chromosome in kangaroos as well. So it looks like that's a very ancient part of the X chromosome, but all the genes in the blue region are in fact on another chromosome. In fact, chromosome 5 in this particular species. And we can actually see this if we take the kangaroo X chromosome, which is rather smaller, and we sort it, and then we make DNA out of it and paint it onto human lymphocytes, and these are the lymphocytes of one of my graduate students. I told him it wouldn't work, but it worked absolutely beautifully. And you can see he's got a single X chromosome, which made him very relieved. And it looks just like my computer-generated picture. So it looks like the human X chromosome has really composed of two evolutionary blocks, one very ancient and one added quite recently. So if I draw that in my usual colours, green for the added region and blue for the conserved region, you can see that by mapping these genes in marsupials, we've been able to look at the evolutionary origin of our X chromosome. Well, what about the Y? When we look at the marsupial Y, it's very tiny, but there's 7 genes on it and they all have copies on the X. So it's just, they're actually different genes, but the same rules that the human X and Y. When we look at the human Y, we get a shock because it's practically all green. So there are only 4 genes that comes from the original Y chromosome that came from the original X, and everything else was added quite recently, and it's probably not very important. So if we look back at our scheme of how the X and Y evolved, we can see that marsupials actually represent a rather final stage in the evolution of the Y when it's completely lost recombination with the X. Well, so we know that the marsupial X and Y are strongly related to placental X and Y, except for this added region. What about going right over to the other side of the phylogeny and looking at reptiles and birds? Do they share the same X chromosomes as well? And because we have a human prejudice, I think everybody thought that they'd all do it the same way. Not at all. If we look at male chickens and female chickens, they do have sex chromosomes, but they're completely the other way round. So it's the male that's got 2 copies of a Z chromosome, which is large and has lots of genes on it. The female's only got 1 copy of that Z chromosome and a pathetic little chromosome that is called the W chromosome, which is female specific. These are completely different from our sex chromosomes because if you map genes that are on the chicken Z, they go to, in humans, they go to chromosome 5 and 9. And if you map genes that are on the human X in a chicken, they go to parts of chromosomes 1 and 4. And this was very nice for us because, in fact, it's the same old evolutionary blocks that we were able to detect looking at marsupials. So the ancient X chromosome is a part of chromosome 4, and the added region, which is exactly the same genes in the same order, is on another chromosome. Well, what about other birds? And this is about the maddest bird in the world. It's called an emu, and it's one of the big flightless birds like an ostrich. And we were very interested in it because it's got funny sex chromosomes. They're Z and W all right, but the W is very big. And in fact, it's almost the same as a Z, as if it didn't really get going on degrading at all. So we looked at the chromosomes, and they're virtually identical to chicken chromosomes except for the Z and W. And what we were able to do is to isolate the chicken Z chromosome and tag it and then paint it onto an emu, and what we found was it's the same Z chromosome, absolutely homologous, and the W chromosome is homologous too, except for a little patch in the middle, and that's presumably where the sex-determining gene is. So what is the sex-determining gene? Well, it's not SRY, because there is no SRY. In fact, the SOX3 gene that we know SRY evolved from, that's also on chromosome 4. So there must be another gene there, and people have been trying to find it for a long time. And it looks now that it's a gene called DMRT1, which is a very interesting gene because, in fact, it has something to do with sex in fruit flies and nematodes as well. So it's one of these really fundamental genes. So it's on the Z chromosome, but it's not on the W chromosome, even in an emu. And very recently, it's been knocked down, that is, made less active, and this causes sex reversal. So you start getting ZZ females because whatever it makes is presumably not enough of it. So this is what we call dosage-sensitive sex determination, and the idea is that you need 2 copies of the gene, both active and making twice as much product in the male as the female. And so it looks like what's happening is you need a certain amount of that Z product, which we now know to be DMRT1, in order to be male. And this seems a bit weird to us because we're used to male-dominant SRY sex, but in fact it's very, very common in reptiles, probably much more common than our way of doing sex. Well, snakes are interesting too, and Australia has lots of snakes. This one's a particularly deadly one. It was one of the most deadly snakes in the world, and this is my brave collaborator. They've got a ZZ-ZW system too, and I always thought it was going to be the same Z. It's not. It's completely different. So it looks like another autosome has been chosen to become a sex chromosome. We've also, quite accidentally, looked at marine turtles, and they're interesting because they don't have sex chromosomes at all. It's actually temperature that does sex. If it's cool, all the eggs hatch into males, and if it's warm, they all hatch into females. So there are no sex chromosomes, but when we looked at the chromosomes and painted them with the chicken Z chromosome, there it is. There's the Z chromosome alive and well, same genes, same DNA, but it's not a sex chromosome because there aren't any sex chromosomes. So the bird Z chromosome is exactly identical to the turtle chromosome 6, and on chromosomes 1 and 4 are exactly the same genes as are on our X chromosome and Y chromosome. So maybe this is what our great-great-great-grandparents looked like. They didn't have sex chromosomes, but they had chromosomes that became sex chromosomes in one lineage or another. So if we look back at our scheme, you can see that a ZW system evolves in exactly the same way as an XY system, but it's all completely back to front. So it's the W chromosome that loses recombination with the Z, and it's the one that degrades. And we now have evidence of just about every stage in this degradation, from the stage where there's no sex chromosomes, like turtles and alligators do the same thing, to the stage where you get very small differences between the sex chromosomes like emus, down to humans which have a small Y chromosome, and kangaroos which have a very tiny little Y. Well, it's interesting to have a look at the bird Z chromosome because we now know it's identical to the turtle chromosome 6, and it's also identical to the top part of chromosome 2 in a snake. But how are we going to know which the ancestral Z chromosome was? Because we can't compare it with anything. Well, very recently, somebody had a look at some gecko lizard, and they found, lo and behold, its Z chromosome is identical to the bird's. And gecko and lizards are not very closely related to birds. So what this means is that we've got now two widely divergent lines with the same ZW, and that has to mean that a ZW system was ancestral to reptiles and birds. So we've got two completely different sex chromosome systems on the two parts of our phylogeny. How did one change into the other, and which one is ancestral? And for that, obviously, we have to look at monotreme mammals. Well, monotremes are the most interesting mammals in the world. Very cute. This is a platypus that you've probably heard of in stories and poems. And they're amazing because, and they're so amazing they actually did a genome of them, because they're like mammals in that they have fur and they feed their young with milk, so they're definitely mammals by any definition, but they retain a lot of reptilian characteristics. For instance, they lay eggs. And you can see on the right there, the hatchlings then lie on the mother's belly and suck milk from her fur. There's milk there, but there are no teats. So they just lick the milk off. And they have other reptile characteristics. For instance, their skeleton is really more like a lizard than a dog or a cat. And they even make venom that's like a snake's venom. So they're very, very interesting, and we thought, well, let's have a look at their chromosomes and pick out the sex chromosomes. So we had a look at the male and the female. We compared them. We should have found one pair that didn't pair up in males, but to our great surprise, we found there are 10 chromosomes that didn't have pairs. So we said, well, that's ridiculous. You can't have 10 sex chromosomes, but you do. There are 5 X's and 5 Y's, and we were able to show this by scraping the chromosomes that were male-specific off a microscope slide, making DNA from it and tagging it green, and then looking at female cells. And you can see it's in the male cell, but not in the female cell. So it's a Y. And then we've got 2 other chromosomes there that we have 2 different paints light them up. And there's 2 copies in females and only 1 in males. So they're both X's. And we're able then to figure out all the X's and all the Y's. So the question was, well, how on earth does it work? How's a poor little... This was from the New York Times when we published this in Nature. So how the poor little baby platypus is supposed to know if it's a male or a female? And what we did then was to have a look at meiosis, the reduction division where the X's and the Y's are sort of supposed to separate, and we found that they were always in an order XY, 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 XY. And what we think is happening, because we never find an X and a Y in the same sperm, is that all the X's go one way and the Y's go another way. So it does work, although it's not the way we would have designed it. So it was really rather amazing to me when somebody told me that our work was featured in the Discovery Institute website. And I said, oh, how nice. And they said, well, maybe you don't understand that this is a creationist website and this is put forward as a wonderful example of intelligent design. So I said, well, that's the dumbest thing I ever heard. And that's been the inspiration for my Dumb Design website through the Academy of Science in Australia. So if you have any good stories for this website, please let me know. So, well, we were very curious, is the sex chromosomes, the same old genes that are on the X chromosome in humans and kangaroos, are they also on the sex chromosomes in a platypus? So we mapped all the genes on the marsupial X, we mapped them in platypus, and they all went at the same chromosome. So that was kind of nice. We've still got the same evolutionary blocks in a platypus, but it's not an X. It's actually chromosome 6. So it has nothing to do with sex at all. So that was a big surprise, and what's more, that explained why we could never find SRY. There is no SRY. In fact, SOX3 is on chromosome 6. So there must be another gene, and we don't know what it is, and there must be other genes on the sex chromosome, and this gave us no clue at all. So then we mapped these sex chromosomes, which took us quite a long time to find out what is on the sex chromosomes, and what we found out is there's no homology to the human X at all, but there's complete homology to the bird Z chromosome. So this was a big surprise. So all those green genes there are on the bird Z chromosome, and what's obviously happened is it started off with a ZW like birds, and it translocated and swapped partners with other chromosomes, so that spread that all over the 10 chromosomes. So what this means then is that monitoring sex chromosomes, and echidna's the same as platypus, are XY, and they're very bizarre, but they're related to the chicken chromosomes, and that has to mean that the ZW system is very, very ancient, and obviously XY somehow or other evolved from that. So now we have ZWs right throughout this phylogeny, and we can see that our XY system is actually rather new. So the SRY gene must have evolved really quite recently, somewhere between 148 and 166 years, a million years ago. So summarising all of that, you can see that when we look at all vertebrates, we can see that there are 3 blocks of genes, the red block and the green block and the blue block. And it's the red block that got chosen to be the ancestral sex chromosomes of reptiles, including the ancestors of mammals. And that's still retained in platypuses. And the green and the blue block have nothing to do with sex at all. But in the rest of the mammals, theory in mammals, the baton has been passed to the blue block. And now the SOX3 gene has evolved into SRY, and the red block has just become an ordinary autosome. The DMRT1 gene is still necessary for sex determination, and in fact, if you delete it, you're a girl. And in humans, the only difference is the green block has been added to both the X and the Y. So we think this is how all the vertebrate sex-determining systems are related to each other. Well, I want to end up talking about the future of sex. And what I've told you, I think, will give you the idea that we know the Y chromosome is degrading, because once upon a time it was just like the X, and now it's very small, and we can calculate how long it's got to go. So when will our Y disappear? We know that 166 million years ago it was just an ordinary autosome in a platypus, and it had 1,340 genes. It's only got 45 today. So we can calculate they're being lost at the rate of 7.8 per million years, and there's only 45, so it doesn't take a genius to realise that it's going to disappear in 5.8 million years. There it goes. Well, the big question, of course, is, will men disappear? And one possibility is, well, maybe the human race will become extinct. Maybe men will disappear. Is that a problem? Could we become parthenogenic? For instance, some lizards are all female, and they make eggs that are just different combinations of the same genes the female has. Could we do that? Well, no, not really, because there's a whole lot of genes that aren't active unless they come through the male lineage. So we have to have sperm, and we have to have men. So these are genes that we call genomically imprinted that are active only if they come through men. So we can't do parthenogenesis, sorry ladies. So our only hope then is that a new sex-determining gene might arise and take over from SRY, and then it won't matter if the Y chromosome disappears. And this will give hope to all the men in the audience. There's actually some rodents that have done this. And one of these rodents is a little vole, a mole vole, that comes from Eastern Europe. And there's 3 species, and one of the species is completely normal. It's got an X and a Y in males, and the Y is much like the mouse Y. But the other 2 are really weird because there is no Y, and there is no SRY. One of these, there's just one X chromosome in each sex, and the other there's 2, but they have nothing to do with sex determination. So we don't know what does sex in a mole vole, but it's not SRY. And presumably there's some other gene that's arisen somewhere else in the genome that's taken over the function from SRY. And we're looking for this very hard. There's a group in Germany that has a colony, and we're trying to track down the sex-determining locus and find out what gene has taken over. Well, could this happen in people? Well, I often think, well, how would we know? Maybe it's already happened. Maybe there are populations in the world, in Patagonia or the Amazon or Siberia, where this has actually happened. Would we know? Well, no, we wouldn't. I mean, mole voles still have babies. They seem to know what to do. There's males and females, and half of the progeny are male and half are female. So if it happened in some population of humans, we might not know unless we actually looked at the chromosomes and thought, hey, no Y chromosome, this is weird. Or if a mole vole man mated with an XX woman. So what happens if a man with a different sex-determining system mated with an XX woman? You would get a clash of the sex-determining systems, which really wouldn't work very well. So I would predict you'd get a lot of intersexes, that is, babies are born that really is not clear whether they're supposed to be male or female, and they're infertile. Now, one thing about infertility like this, it would instantly impose a reproductive barrier between these populations, and that's exactly what you would expect for a speciating event. So if you come back in 5.8 million years, you might find no humans, or you might find 2 or 3 different hominid species. So I don't know which is the scarier thought. And if you think I'm exaggerating, this is exactly what happened with mole voles. So I'd like to make some conclusions from all our studies on weird animals and weird vertebrates, and my conclusions are that sex chromosomes really play by some very strange rules that are quite special to sex chromosomes. But they're very general. They're the same rules in a human and a bird, and probably a skink and a turtle as well. We conclude that our XY system is actually rather recently evolved. It was still an autosome just 166 million years ago, and that means it's degrading very rapidly, more rapidly than we thought, and it will eventually disappear, and we can argue about how long it's got to go, but eventually it will disappear. But a new sex chromosome system might evolve and save us all from extinction. So the subliminal message again is that you can learn a lot from weird animals, questions you didn't think to ask, and answers that are quite different from the answers you get in humans and mice. So lastly, I just want to thank many generations of students and some postdocs in my lab, and here's my Sex and the Y team all lined up in my university at the Australian National University, and I've depended very much also on collaborators who know about turtles or frogs or so forth, and also I've had a lot of very good interactions with some friendly genome centres who sequence some of our genomes that the Australian government's too mean or too poor to do. And this is something that you may have seen in Time magazine many years ago, is the gene loci on the Y chromosome. And I always thought, well, we really need an X chromosome to go with it. So over many years, I've developed some X chromosome loci. Oddly enough, I can't find anybody who wants to publish this. But I'll leave it up for you to enjoy while I answer your questions. Thank you.
My wife definitely wanted to come to this lecture, and I think she's learned too much. We do have an opportunity for a few questions. There are microphones in the 2 aisles, and let me suggest that anyone who has a question, if you'll come to the microphone, we'll try to cover at least some of them.
Could you explain why some species can change their sex in the process so as to maintain a balance of sex in that species?
Well, that's a very, very interesting situation, and there are many species, as I'm sure you're aware, where the hatchlings will first of all be females and then later in life become males, or the other way around. So you can ring in practically any sex ratio you like. It doesn't have to be 50-50, and in fact in many reptiles it's very far off 50-50. For instance, that you really only need one male and lots of females in order to get a very large second generation. And that's thought to be the reason why temperature-sensitive sex determination evolved, because, for instance, in alligators, I believe that when there are not many alligators around, the females prefer to build their nests down by the water where it's cool, so these are female-producing nests. But when the population pressure builds up, then they have to, some of them will have to build their nests up on the levees where it's hot, and so you start producing males. So it seems to be a bit of a population control mechanism in alligators. But there are fish, for instance, that change sex. If you, for instance, the blue wrasse, if you take a male out of the tank and there's only females there, then the biggest female will become a male very quickly, in about a week. So it's absolutely amazing that there seem to be all these males in waiting. But of course, it's better than fighting to the death. You're not wasting all these males. They're good females and they're laying eggs. And so you can see why that system might be very selective.
Could you comment on the fact that the Y chromosome is, as I understand, a palindrome, and whether that also applies in the Z's and W's?
Yes, in fact, I eliminated this slide, so thank you very much for asking that question. But the Y chromosome has a very strange structure that's been elaborated in David Page's lab. A lot of the genes on the Y chromosome have many, many copies. And it turns out that these copies are very often in long stretches of DNA that are what we call palindromic. That is, they read forwards and backwards on different sides of the arms, so that they can actually pair. And it's been claimed that this pairing will save the Y chromosome because it can't pair with the X anymore, and it can't swap bits with the X to make it whole again, but maybe you can correct mutations by reference to the gene on the other side. But in fact, I don't believe this. And for instance, if you look at the bottom loop has got a group of genes called DAS genes on it. If you get a mutation in one of them, it could be repaired by the other one. And so, you know, you're saved, you haven't lost this DAS gene. But it doesn't work like that, because you could equally well get the other way around. So in fact, it's the mutated gene may make a copy of itself. So I think it's just going to change the way that the Y chromosome degrades. It's going to be much jerkier than we thought it was, but I don't really see how it's going to save the Y. The other thing is that we know that, for instance, the RBM gene there has got 28 copies, but only 3 of them are active. So 25 of them have gone, and it may be that this mechanism, which I call doubles or quits, has got rid of them preferentially. So even very, very useful genes like these ones that we know have a function do drop out in some lineage or another, and I don't think being in a palindrome is going to save it in the long term.
Do the Z's or W's have palindromes?
We don't know yet, but there is work being done on the chicken W, and I'll be fascinated to find out if there are palindromes. There are palindromes on the chimpanzee Y, but they're actually different. So it's the same palindromes, but they might be shorter or longer, but there's also some completely different palindromes. And of course, we're trying to sequence fully the marsupial Y to see that has palindromes as well. It may be a function of the Y chromosome not being able to repair itself that is subject to the insertion of all this junk, a lot of which is repetitive sequences, and so it may be particularly vulnerable to this kind of amplification and deletion.
Please.
May I ask you to elaborate on the role of imprinting in gender identity, specifically the role of imprinting via methylation, and is the concept still that methylation-dependent imprinting is not in evidence in monotremes and in lower mammals, and also if you could comment on the role of retrotransposons as part of these brains and balls genes?
Well, that's a very interesting question. We could talk about it all day. But the first part of it is are imprinted genes important in sex determination? Now, I mentioned imprinting, and we now know that there's at least 100 genes that are only active if they come through the egg, and there's 30 or 40 genes that are only active if they come through the sperm, and that's called genomic imprinting. And the main role of sex chromosomes, and in fact this is what got me started on sex chromosomes in the first place, is because the X chromosome is imprinted. It's completely inactive. There's 2 X chromosomes in females, and one of them is not active. And that is the best example of epigenetic imprinting. And in marsupials, it's always the father's X chromosome which is inactive. And so that was actually the first evidence that imprinting occurred in mammals. Whether it imprinting is important in sex determination, we don't know. I don't know of any work that would suggest that, but it certainly is very important in dosage compensation of the X chromosome. The retrotransposon question, again, we really don't have a terribly good idea what these retrotransposons do. They seem to be largely dead viruses that got left in the genome, and they accumulate in both the X and the Y because of the peculiar structure of the X and the Y and the fact that there's only one of them. So that seems to be why they're there. However, once they're there, they can start evolving functions, and we know several that have now evolved a function in regulating other genes.
Yeah, last question.
Are idiots savant mainly male, and if so, is it due to a defect in the Y chromosome?
Yes, they are mainly male, and there's been a huge hunt for autism genes. And I heard just yesterday from somebody who studies them that there do seem to be genes that are associated with autism. They're not on the Y chromosome, however. There was a suspicion that there might be a schizophrenia gene on the Y, but I haven't heard anything about that. I think it's sort of disappeared. But most of the genes on the Y chromosome seem to have everything to do with making sperm or making a male. Very few of them seem to be expressed in anything except testis. And the ones that are probably just were put on the Y chromosome quite recently and haven't had a chance to get off it yet.
Please join me in thanking Jenny Graves for having provided us with a wonderful evening. Thank you all for joining us.